From the floors of the factory to the farmer in the field, Newport-Fersan, Jamaica is committed to building and maintaining a strong manufacturing sector. We know fertilizers, and we know you. That is why we have been partnering with farmers to provide improved blends, coffee-lizer, Ferti-Peanut, potato-lizer, and more. Manufacturing is a creator of jobs. It is a driver of the Jamaican economy, building strong community ties and stimulating innovative thinking and creativity. And so from the committed men and women who work in our factory, to the members of our management team, to our agronomists and everyone in between, we are all committed to maintaining the highest standards of manufacturing. And we have seen this commitment by attaining the quality standard ISO 9001-2015. Newport-Fersan, Jamaica Limited, the first on the land, committed to manufacturing, committed to you. From the floor.
What's in store this evening? All right, so session A, Mr. Melvin Henry, background justification, what are plant viruses, process of viral infection, general symptoms, modes of transmission, and then you will have Mr. Sajae Jones, who will be discussing who will be taking a look at the most common plant viruses, identification of viral symptoms on select plants, management strategies for plant viruses, an in-depth look at FERSAN STAR Virucide, benefits to be dried from STAR Virucide, and high-yield package cocktail. So we have a very exciting session for you this evening.
Okay, thank you, Latisha. Good evening, participants. Are you hearing me?
Yes, sir. Yes, sir.
Good. Okay, so as was outlined, okay. Let me first take this opportunity to say thank you for taking the time out of your busy schedule to participate in this evening's activity. And as Latisha said, the training will be segmented into two parts, a part A and a part B. Okay, so if it is that we would have invited you here this evening, it's only fair that we provide you with some justification why we're taking some of your time to look at the whole issue of viruses in agricultural crop production or in agriculture in general. Now, according to the UN FAO, Food and Agricultural Organization, FAO statistics, it is dictated that by 2050, it would be necessary for us as farmers to produce 70% more food than we're currently doing, because it is estimated that the world's population will be around 9 billion strong. And the graph you're seeing actually highlights the progress as it relates to us moving towards 2050. So that in and of itself says a number of things. And one of them is, for me, is that the whole business of agriculture will forever remain alive and well once human beings exist. And therefore, there are opportunities. It also means that if it is that we're going to increase our output or productivity, our production really, by 70%, it means that all the variances and all the factors that could negatively impact production and productivity should be on our radar as operators within the sector And as such, steps must be taken if it is that we're going to be successful. With that said, another well-documented set of information is that an estimated 30 billion US dollars in economic crop losses is what is achieved or obtained or realized annually, 30 billion US dollars. And we're talking about just plant viruses here. We're not referring to the other factors or the other diseases or biotic factors or abiotic factors that contribute to losses. We're only looking at viruses, and this is what the data states. Additionally, what the data is suggesting or stating clearly is that viruses are linked to 50% of plant diseases worldwide. Now, that is significant. I just mentioned that there are a number of other factors that contribute to plant diseases. But if in reality, 50% of these plant diseases are actually attributed to the actions of viruses, then that in and of itself is a serious concern as it relates to the whole issue of global food security. So with that said, this bit of information actually forms the background, yes, for us actually starting this sort of a discussion this evening. So what are plant viruses? So plant viruses, as the name suggests, would naturally be viruses that are specific in their attack or affecting plants. It is said that plant viruses are microscopic, meaning that they cannot be seen with the naked eye and will require an electronic microscope to see them. Now, the viruses actually compose of different parts, and we would have gone through from 2019 to now, actually weekly hearing the term, albeit we're referring to a virus that would have been affecting human beings. But the principle is similar. It is essentially composed of nucleic acid and a coat. And as you can see by the graphics below, this is pretty much the structural look of the viruses. Now, it is very important to note that they are referred to as intracellular, obligate intracellular parasites. And what that means is that by themselves, viruses cannot replicate, form new ones. So what they depend on is being in the cell of a host plant, right? So once they get within the cell of a plant, that is where they are able to, that is when they are able to replicate and cause serious damage to the organism in which they have invaded. It is said that they are also pathogenic or disease-causing to higher plants. So that's the main concern, and the data that we looked at just now as it relates to the percentage of plant diseases that are linked to plant viruses, which is 50%, supports that claim. Now, in order for us to have an infection, there has to be a particular approach or a sequence of activities that happens. Now, once a plant is infected with a virus, meaning the virus enters the system of the plant, it immediately starts to effect changes within the plant. So it starts to effect, at first, structural changes. And then once those structural changes happen within the system of the plant, then you will also see what we refer to as physiological changes. And I'll explain just now. So let us look at the chart on screen. The virus, upon infecting the host, starts off as a single particle. Then upon entry and finding itself into the relevant cells that will facilitate it developing and causing problems within the organism. And in this sense, we're looking at plants. At stage 3, it starts to replicate. So we started with 1. And if you can look here, you're now seeing more than what started. Because at this point in time, the virus actually starts to replicate, and you're having now an expansion, if you may, of the population within the system of the plant. Now, the more this replication takes place within the system of the plant, and might I say that no segment of the plant is spared once this process of replication starts. So quickly, it will spread throughout your foliage, it will spread to fruits, it will spread to tubers below ground, your entire root system, because there's no specialization in where this replication and transmission will actually take place. So the net result is the longer this virus remains within the system of the plant, then the more the chances for replication and the more that infected or infested plant pose a threat to plants that are not yet infected. And we'll get into how the transmission of viruses from the infected to the uninfected plant occurs later on. So this is pretty much a cross-section of a specialized tissue. If you look at the picture within plants, and this is actually the system that transport, be it nutrients and water, in what is referred to as the xylem vessel, or the products of photosynthesis in the phloem. So once you produce food, once food is produced within the plant, then there's need for this to move to the storage organ. So let's say we're looking at the Irish potato plant. An Irish potato plant, through photosynthesis within the leaves, produces food, but this food must be sent from what is called the source, being the leaves of production, to the sink, which are the tubers below ground. And the transport of these foods to develop the tubers below ground actually takes place within the phloem vessels, which are to the extreme right of screen. Now, essentially what happens when a virus enters the plant system is that, as I said before, it starts to make what we call structural alteration. So once you had free flow of water and nutrients within your vessels, the xylem vessels, now that becomes impaired, because one of the actions of viruses is that they start to clog the transport system. And as a result, once that starts to happen, you will notice changes. So bottom of screen, we have two main types of changes or alterations taking place within the plant. One, referred to as morphological, which simply put is the form, the shape, and the structure of the plant itself, be it the root, the stems, the fruit, or the leaves. And then once that is happening, you will see what we call physiological changes, meaning the functions and processes within the plants are now compromised because of the morphological changes. And let me use an analogy. Imagine a garden hose. It is used to fetch water from the top to the garden. The shape, the form, and the structure of the hose, which is the morphology, allows the water to flow seamlessly through the garden hose from the top of the pipe to the garden to facilitate plants growing. Now, So the function is to deliver. That is the whole process. Now, once there is a twist, there's a crimp on the hose, what you find happening is that this garden hose no longer allows the free flow, if any at all, of water from the top up to the garden. And this is how now you start to see the effects of that crink. So the crink in this scenario would be the actions or the action of the virus within the system of the plant. And once that happens, it causes significant changes. And those changes will pronounce or provide symptoms that we use to determine that something has gone awry or wrong with the entire system of the plant. So before we move further, I'd just like us to look at a few key definitions. One, disease. So a disease really is a disruption of the normal processes, and it is normally by a causal agent. Now, it is important to note that a disease can be classified as infectious, meaning that it is caused by a living organism, a biotic agent, and it means that this disease is transferable. A disease can also be classified as non-infectious, meaning that it is caused by an agent that is not biotic, but abiotic. So let's say the pH within the soil or a nutrient deficiency. Those also are results in diseases, but are not infectious, but non-infectious, and those cannot be spread from plant to plant. So once a plant is diseased, then there's the disruption of the structure, the normal structure of the plant, and the growth and overall function and other activities within the plant system. Now, in the entire sphere of being a productive farmer or operator, it is very important to note that the implication of a disease on a plant is one of such that it could have severe implications on your thrust to have the best possible yields in your production plan. So this is pretty much an example of a cyst, a nematode cyst on the root system of a plant. Now, there are two other terms that we must make some distinction between: signs and symptoms. Now, signs refer to the physical evidence, in this case, talking about disease of a pathogen. So for example, you're able to see fungal spores, fruiting bodies, or the oozing of bacteria from the tissue of a plant, or as we have it here, the nematode cyst. So those are signs indicating that something is happening. And these actually play a critical role in us identifying diseases. Now, symptoms, on the other hand, are visible effects of a disease on a plant. So there are signs where you can actually see the causal agent or its action, and the symptoms really speak to the visible effects. Examples include changes in color, shape, and or function of plant in response to the invasion of a pathogen. All right, so there is some difference between, so these terms are very important for us to understand as we progress in understanding how the disease occurs. Now, as it relates to viral symptoms, there are some general descriptions or characteristics of viral infections, and it is very useful for us to familiarize ourselves with some of them, so we can have an insight as to what is happening in our fields. Be aware that these general symptoms are not specific to any particular crop. And as such, they can be seen on any crop in question. So the first one that we look at as a general symptom is what we refer to as a mosaic. And this is really characterized by a mixture of light green and yellow patches on the leaves. And that is what we actually refer to as the mosaic pattern. So if you look at the leaves below, to the extreme left, the papaya leaf, you're seeing that light green and yellow, and that is an expression of the symptoms of a mosaic disease affecting the papaya plant. On the right of screen, we have the watermelon mosaic virus affecting the leaf. And again, you're seeing those variations in color. It is also important to note that sources of infection in these cases are normally systemic, meaning that they actually find themselves within the system of the plant. And the examples of other mosaic diseases include tobacco mosaic, mosaic of cucurbits, mosaic of potato, and also sugarcane suffers from this mosaic pattern. But the real question is, what are the implications of this mosaicing or mosaic pattern on the leaves? Now, in order for the plant to produce at its maximum capacity, it requires a healthy leaf. Now, if the plant leaf is not healthy, what it means is that it will not function as it should. And that in turn will have serious consequences for how well that particular plant is able to produce. Remember that the leaves of the plant are the major manufacturing centers. So you cannot have a profuse flowering, fruiting, and the development of fruit if it is that the quality of your leaves are impaired or substandard, okay? So that is why mosaic is something that you should have an eye out for and take the necessary steps, which we'll look at later on in the presentation. Another set of general symptoms has to do with uniform yellowing or chlorosis of the leaf. And this is an example seen in the rice yellow mottle virus. So what you have is this general chlorosis. And again, if the leaf is not green as it should be, the yellowing is an indication that those cells are not up to par. They are not ready to manufacture. And as a result, in this case, this rice field would not be able to produce as it should, because there is the absence of the necessary level of chlorophyll within the leaves to enable it to capture sunlight and execute the normal functions within the plant. Another general symptom, once there's a viral infection, is what is referred to as vein clearing. So if you look to the right of screen, you'll notice the leaf of an okra plant. And what you're seeing is that the veins and veinlets becomes yellow while the rest of the leaf remains green. Now, what this is actually indicated, and it is of serious concern, is that a lot of your major transport vessels actually passes through these veins. So if there is something impairing the transport of water and the nutrient within the system of the leaf, then ultimately the productive capacity, the ability of the leaves to really produce, to facilitate flowering, fruiting, and the development of fruit or tubers below ground, rhizomes, you name it, the productive part of the plant then this would be seriously compromised and becomes a serious issue. Another symptom, general symptom, is what we refer to as vein banding. Now, this is pretty much the opposite of vein clearing. All right, so the entire leaf turn yellow and the veins would remain green. Again, the consequences or the consequence, one of the consequences of that is that your leaf is now not in its best state to produce. It's not able to capture sunlight and convert into making food. And as a result of that, your field or your plant becomes less productive than it genetically should be performing. Leaf curling and the leaf rolling are also two common symptoms once a plant is infested or infected with a virus. So some of the common leaf curl that we have are the leaf curl of papaya, leaf curl of tomatoes, and the leaf roll is different from a leaf curl. So if you notice, a leaf curl does not necessarily lead to the margin or the edges of the leaves, both sides of the leaves coming together. Once you have a leaf roll, as is shown on screen with the potato leaf roll disease, you actually have a situation where the margins form like a tube, right? Now, once this happens, it actually creates a number or set in motion a number of challenges. One, the leaf is not positioned to capture adequate sunlight to facilitate photosynthesis. And as a result, this potato plant would not be as productive as another plant that is not infected with the potato leaf roll virus, thus giving the disease. Another problem that you could find here is that because of this rolling of the leaf, it becomes an haven for some insect pests. So they find themselves beneath because there now is some amount of shading, and there you have a secondary problem. So that is really those are really some of the consequences of having the leaf roll disease in the field. And once you're having either the leaf curl or the leaf roll, they're normally followed by some signs of mosaic, where you start to see some yellowing of the leaves as well. So it is important to note that a single plant can be infected with more than one virus at the same time. A little leaf symptom, as the name suggests, because of the impairment or the blockage of your transport systems, the regular size of your leaves are not realized because they're having restrictions in accessing the requisite volume or quantity of nutrient and water to facilitate development. And as a result, instead what you have is a distorted rosette-like structure forming similar look to a rose. The leaves are clustered. And this is something that is very, very common in sweet pepper. That one stands out easily in sweet pepper and affects tomatoes as well and other crops. Stunting is another general symptom, and simply put, it's that retardation of growth. So the plant is not developing at its usual rate. And oftentimes we tend to point the finger or guess what is transpiring, why it is that the plant is not growing as it genetically should, or others within the field are growing. And this is actually one of the reasons if the plant is infected with a virus, this could be one of the results. And as you see on screen, once that stunted growth takes place, it simply means that other ailments, if you may, will follow. And as a result of that, your quest to have your crop producing at its maximum become severely impaired, and it's actually a downhill if corrective measures are not employed. So we would have looked at the symptoms, we would have looked at what a virus is in principle. So it's very important for us to understand how it is that viruses are transmitted. Now, as I said at the outset, viruses are referred to as obligate intercellular parasites. They live or they benefit from living within the cells. That's the only way they can replicate. Now, once you have an unhealthy plant within the field, to the extent of the infection, it is a high possibility that you'll have transmission if the factors or the modes or the different ways in which the viruses are transmitted comes alive. So we're going to look at transmission by vectors, namely insects, mites, nematodes, and fungus. We're going to look at transmission via vegetative propagation, mechanical transmission via plant sap, transmission via seeds, pollen, and also by way of soils. So as it is, the transmission by insects is by and far one of the most common ways in which viruses are transmitted between plants. Now, based on the very feeding actions of insects, it facilitates them picking up the virus from the sap of the plant, be it a chewing, rasping, or sucking pattern when they're feeding. And then once they move to another plant, which may be uninfected, then that is when you have that transmission taking place. It is important to note that aphids are known to be the #1 vectors that transmit viruses. And a vector, simply put, is an organism that transmits virus in this case. Now, whilst the picture depicts an aphid being green, be assured that there are several variations in color. Dark brown, light brown, there are several appearances. All in all, they are notorious as it relates, the most important set, followed by leaf hoppers, mites, thrips, and whiteflies. So these vectors are of serious concern. And in reality, the most dangerous implication for having these insect pests within the farm is not so much just the fact that they're feeding, is the fact that they can do a greater damage by transmitting a virus. So pretty much the same principle as it relates to mosquitoes. It's not the bite that we're really concerned about. It's really what happens after that bite, right? So that transmission of virus is really what set in motion the challenges that the system of the plant will have to deal with thereafter. So based on how the transmission takes place, the transmission can be referred to as a persistent in terms of how the virus is transmitted, meaning that it lives within the insect's body. It becomes a part of the insect's metabolism. It is transmitted from parent to offspring, meaning that if that insect reproduces, it simply means that the offsprings as well will have the viral particles within the young. Right? So it's very important to note that in this situation, they actually become partners or body friends for a lifetime because of the mode of them actually entering the system of the plant and residing there. Now, the non-persistent transmission, the virus is retained into the stylets, for example, the potiviruses. And the stylet pretty much, once the organism feeds, it activates an interaction between the vector, the plant, and the virus itself. And it is once that feeding takes place that the virus is no longer a part and parcel of the system. So there's a difference between it staying within, residing within the system, and it being left. And that is why oftentimes we normally advise farmers, for example, doing, let's say, hot peppers, to use a barrier crop. So if you plant corn, not so much for the economic gains, if you have a non-persistent virus being transmitted and the organism, the vector, stops on the corn first to feed, and then proceed to the pepper field, what you'll find happening is that it will not enter the field with a non-persistent virus. It would have left it on the corns at the borders. Now, this is pretty much just a close-up shot of what transpires as it relates to the organism acquiring the virus from an infected plant, and also at the same time, the arrow is showing how it also transmits in both directions. So it either is receiving or picking up the virus, or letting or infecting the plant through a process we call inoculation. So that's pretty much how that process gets started. This is pretty much a graphic which show persistent versus non-persistent, the virus having entered the system of the vector. If it is persistent, it remains and is transmitted throughout the entire system of the plant and resides there, while in the non-persistent, as explained just now, it would have left it once it feeds. Mites, which is another classification of pests that affects us a lot, namely, and some of the major ones are the red palm mite, fraud mite, spider mites. It is said that at least 6 viruses are transmitted by mites, and this is significant. And you're seeing where, for example, those mosaic type viruses, viral diseases, are actually the causal agent in a lot of cases are actually mites. Some mites are also referred to as a non-specific causal agent for the potato virus. So we have a cause for concern once there is a mite infestation in your potato fields, because the reality is that separate and apart from them being there, you could very well have another problem where they're transmitting viruses, and that will compromise the productivity of your crop. Transmission also takes place via nematodes, and these are worm-like organisms that normally colonize or live within the soil. and then seek to inhabit the root system of the plant. Now, if it is that the nematode upon entry to one plant system that is infected, an infected root system, and then makes its way, if it's one of those that move, because you have some of them that are classified as sedentary, meaning they stay one place. The reality is that you have a high chance of transferring because of the interaction with the sap from the cell, the root cell, and then the chances of you transmitting this virus to another plant is a high possibility within the soil. So for example, the grape, fan leaf, and tobacco ring spot viruses are normally transmitted in a number of cases by this modality, by nematodes. Fungus actually are not to be spared or left out in the fact that they too aids in the transmission of viruses. And it is said that at least 30 plant viruses are transmitted by these three main groups. And one you can see bottom of screen is the Spongospora species on the potato. So not only is it distorting the look and making the potato unmarketable, but the reality is that once it infests the system, then it means that even the procurement or securing of seeds for a subsequent crop is in serious state, because the virus will reside within the tuber because the tuber is alive. And for your subsequent crop, it simply means that you'll have not only an inoculation or spreading to a new production area, but the chances of your plant being as productive as they should becomes thin. Transmission via vegetative propagation, so budding, cuttings, grafting, and ear layering are some of the means. So if it is that you're moving infected parts or propagation material to another area, that's also another way where we have the transmission. Might I hasten to say that sweet potatoes have been suffering and having some serious concern as it relates to viral loads. And normally, some crops do not display as pronounced as others the symptoms. And you could very well have a situation during COVID time we were talking about, we're using terms like asymptomatic, meaning a person could very well have within the virus, but not exhibiting any symptoms. But be assured that once the system of the plant is affected with a virus, you could very well see a net result being lost in the productivity of the plant. And we would have seen this time and time again that, for example, you might very well have, this could be one of the causes, you could very well have a sweet potato field that looks normal to the eye, but at the time of harvesting, you realize that what you're finding below surface in terms of root tubers are less than to be desired. So I know, for example, that locally the ministry has been doing a lot of work in providing clean planting material. And this is one of the reasons why such an effort has been undertaken by the Ministry of Agriculture. Again, tubers, corms, bulbs, and rhizomes are sources of transmission, because once the virus particles, the viral particles are found within these, and you start to do propagation, it simply means that you are at risk for your subsequent crop establishment to be impacted in some shape or form by the presence of these viral particles. Mechanical transmission, there are times when the plants have abrasions because of probably the activity of wind. And once that happens, if two plants are in close proximity, one infected and one not, but once you have the wounds being formed and you can have the exchange of plant sap, then that's also one such way in which you have that happen. You could have it, as I said before, again, mechanically by cultural activities or you doing propagating processes such as budding, you're using knives and stuff like those. Those are actually sourced as well and are referred to as mechanical transmission. So once there is the exchange or the passing of plant sap, it's a high possibility that you will have the transmission of virus from one infected to an uninfected plant. So again, handling is also one of them. So the curly top virus in cucumber mosaic virus and cucumber mosaic virus are spread via handling as well, because there are times, for example, as farmers, you're harvesting your ants from time to time, come in contact with the sap from a particular plant and you move to another. And so the likelihood of transmission via that modality is also very likely. Transmission via seed. So it is said that approximately 100 plant viruses are transmitted via seeds. Apologies for the typo there. Only an estimated 30% of the seed derived from virus-infected plants transmit the virus. And this is actually a situation on average, because as you talk about different viruses, the percentage varies. So this is pretty much an average. And you would appreciate that 30% of your planting material in the form of seeds being infected with a particular virus would be of serious concern. Because once you establish with the other 70%, then it's just a matter of having the vectors or the other modalities coming alive for you to have the other 70% becoming infected with the presence from this 30%. Now, as it relates to the tobacco ring spot virus affecting soybean, it is said that nearly all, 100%. So we just talk about averages, but for this particular virus, the tobacco ring spot virus, which affects, for example, the soybean plants, in this plant, what you'll generally find happening is because the ease of transmission throughout the system of the soybean plant, you will see that the seeds, so you will see later on when we look at management practices, in a situation like this, it would be advised to secure planting material from such a field. Now, the basic structure of a seed, and those are, as they are, plumule, radicle, the cotyledon, the seed coats, the embryo. And be reminded that the seed is alive, and it therefore means that there is the possibility and the likelihood of the viral particle surviving within your seeds. Transmission via pollination. Now, pollination is a very critical process for us to achieve production in our flowering plants. Now, because we have the possibility of experiencing what we call cross-pollination, one pollen moving from one plant to the stigma of another plant, the transfer of the viral particles in pollen is highly likely. And this is also one of the modalities or ways in which you have this sort of a transfer. And this is something that we should be mindful of. So once there's a presence of virus, looking at the different modalities of or means of transmission, you can see clearly that there is serious cause for concern, and every effort must be taken to see how best we can curtail or manage the happenings. Just to point out that in terms of transmission, transmission could be referred to as vertical or horizontal transmission. Now, vertical transmission means that the pollen is transmitted from adult or parent to offspring, right? So we're talking about seed, if we're talking about suckers. So you're talking about moving from one generation to the next generation. Now, horizontal transmission happens where plants are of the same generation. And just like we depicted just now, you're having mature pollens being transmitted to or transferred to the system of a plant in close proximity, or by the assistance of a pollinator, for example, the bee. Okay, so that's it as it relates to the virus itself, how it infects the plant, its transmission. And at this point in time, I'll hand over to Mr. Sajae Jones, agronomist, to take us to section B of tonight's proceeding.
Good evening, farmers. I just asked that someone confirms that they're seeing my screen.
Yes, Mr. Jones.
Thank you very much. Okay, thank you, Mr. Henry. So to continue with our presentation on viruses this evening, we just take a look at some of the most common viruses of agricultural importance. To start things off, the tomato mosaic virus. As you would have heard before, mosaicing has to do with a yellow or a yellow-green discoloration of the leaves. You can almost think of it as a liver spot-like look. So the potato leaf roll is another. Papaya mosaic virus. I know whenever papaya farmers hear about this, they start to cringe. No one really wants to hear of that. The citrus tristeza virus, the chili leaf curl, the banana bunchy top, the tobacco mosaic virus, the tomato spotted wilt virus, the tomato yellow leaf curl virus, the cucumber mosaic virus, potato virus Y and X, cauliflower mosaic virus, African cassava mosaic virus, plum pox spotty virus, broom mosaic virus. And there are lots more that could be in this list. Now, it is important to note that viruses does not only affect fruit or vegetable crops, but they also affect ornamentals. So It affects all plant life. So it's not subjected to one category, but all the categories can and are affected by viruses, or can have a viral infection that manifests itself in some ways that we don't want to see. For example, your orchid here. Some signs of infection of viruses in orchids can be a mosaic pattern. The flowers will have deformities and the color breaking. Shrunken chorotic or necrotic patches can be found on the leaves or the stem. And here we can see an image here. So image A to your left would show mosaic patterns on the leaves. So if you look close, you can see that discoloration right there. Or on your right now, in image B, you'll see flower with deformities. So up here, you'll see where you have the nice flower, and here you'll see no flower because it's deformed, it's broken off. And that's caused by viral infection. Here you can see the necrotic patches and sunken patches on the stems and on the leaf of the plant. You can also have viral infection in a watermelon crop. And I know a lot of people are joining close now to their screens and saying, watermelon, all the years we've been battling some problems. Well, now we're here with a solution for it. So you can see sometimes you go out in the field and you see the vines turning upwards, or you can see a bleach look on the watermelon, a mosaic look on the watermelon leaf. As you say, it's a liver spot-like look. Some of the symptoms can include fruit abortion or uneven ripening, internal yellowing or the breakdown of the flesh. Infection may also cause fruit malformations. So sometimes you go out there and they don't have that oblong shape in the melon. It can cause stunted growth as well. To your right, you can see the mosaic-like pattern on a watermelon leaf. So you'll see there, you see the yellowing in it, and that pattern, wherever you see that, it's a sign of a mosaic, the cucumber mosaic virus to be more specific, in watermelon. On your right, you'll see an image of the vines turning upwards in the watermelon field. That's another sign of viral infection in a watermelon crop. Here, if you look close, you can see that the fruit, right here, you can see the malformation of the fruit right here, and you can see the uneven ripening of the fruit in the watermelon here. So those are signs of viral infection in the watermelon. So we can also have, as you said before, viruses can infect any plant. Here we can see some of the infection, some of the signs of virus in your tomato crop, or a yellow or a white or a green stripes, streaks or spot on the foliage. So that's another way of telling about the mosaic virus. So if you see those, you can think about tobacco mosaic virus, which affects solanaceae. So your tobacco, your tomato, your Irish potato, tobacco mosaic virus can affect it. You can have wrinkled or curled leaves, small leaves, pronounced yellowing only of the veins, stunted growth and reduced yield. Infected fruits can appear mottled and can have raised warty areas. You can see that happening here in the image to your left, where you have the warty areas on the fruit right there. So this right here is a sign of the tobacco mosaic virus in your tomato crop. You can also look right here to your right and see where the leaves have that mosaic pattern in it. So wherever you see this on the tomato leaves, you know that it is a high possibility that it is infected. It is infected with the tobacco mosaic virus. Symptoms of the mosaic virus in papaya. So you'll see severe mosaic or mottled leaf. Again, we can describe it as a liver spot-like look. So you can see that in these images here, you can see the mottling of the leaves, mosaic pattern on the leaves here. So if you see a papaya leaves having these discolorations in them, you know that it's a mosaic virus causing it. Another virus that can affect your papaya that all papaya farmers fear is the papaya ring spot virus. If you approach a papaya farmer and ask him what ails him, he will tell you that it is the Papa ring spot virus that ails him. Hand on his head, you know what's bothering his brain. So you look for the yellowing of the leaf distortion, or oily water-soaked spots or streak may appear on the trunk and the petioles. The fruit will have bumps and classic ring spot. In severe cases, you can have necrosis, meaning the fruit can be rotting or the tree can be rotting. You can look for the shoestring distortion of the leaves. So you have leaf curling, you have leaf rolling, and this is now the shoestring. This is another sign of a viral infection, and it's akin to the Papa ring spot virus. So you can see here where the leaves become thin and slender. Yes, that's the Papa ring spot virus presenting itself right there. Here, if you look closely on the images to your left and right, you'll see right here have that water-soaked look on the trunk. Right here, if you can see where the laser pointer is, this is a sign of the Papa ring spot virus. This is how you can identify it in your papas. You can see it right here again. Papa ring spot virus also presents itself on the fruit. We have these rings on the fruit. So you can see an example here with an unripened fruit, and then on your right, you see a ripened fruit. So you can have a good visual look at what it is. This is how you identify the Papa ring spot virus. Preventive measures for your viral infection. So you're going to want to avoid exporting or importing any disease planting material. So you don't want to import it from anywhere that has the Papa ring spot virus, for example, the tobacco mosaic virus. You don't want to inspect any planting material that has that on it. And you don't want to be exporting it again. You don't want to be the one putting more virally infected planting materials out there. You want to select viral disease-free seeds from disease-free regions. So again, this is tying to the import and export. Select disease-free planting materials. I know that Borders does that. So you want to select cuttings, rhizomes, and tubers that are free of disease. Cultivate trap crops that will avoid the disease-causing insect vectors. So as Mr. Henry explained earlier on, you want to plant up your border crops so that the non-persistent virus can stay on that. As the vector comes in, it will land on that crop first and leave the virus with that crop and not take it into your crop that you want to have agronomist have your main crop of focus, that is. Application of soil fumigation for nematodes to transmit the virus, because of course, if you get rid of the vector, then there's nothing to move the virus. Destruction of weeds that serve as hosts for virus. So you don't want an instance where your field was infected with virus and the field is no longer there, but there is a host plant there for the virus. So it just sits and waits until you plant again. You don't want that happening. So you want to remove your weeds that can host the virus in the interim. Cultivation of resistant varieties of plants. Application of temperature extremes. This can be found in, for example, sugarcane, mosaic can be destroyed or reduced by hot water treatment of 52 degrees Celsius for 30 minutes. So it's a way of removing or destroying your viral infection in sugarcane mosaic. Application of insecticides to control the vectors that serve as hosts. So again, if you remove your vectors, then the likelihood of the virus being transmitted is reduced significantly. So if you remove your insects from your plot, then you won't have anything to transmit the virus from outside of your plot or for spreading the virus inside your plot. Now, management of virus using the STAR. Now, I know this is the part that everyone's interested in. What is the STAR and how can it help me? How can it improve my operation? How can it turn around something for me? Well, STAR is designed to enter the stomata's open and translocate into the plant system through the vascular bundles. So on every leaf you have stomatas, right? Those are small openings, and that's where the STAR enters through. The STAR is a broad spectrum virucide. It protects the plant from viruses and improves the resistance if infected. So it being broad spectrum means that it covers a wide range of virus. So every virus that you can think of now that is affecting you, the STAR can aid you in that. It contains active ingredient, plant extract consortia, which is a combination of several unique plant extracts. And this is how it goes about being a broad spectrum virucide. So this is how the STAR really works to control plant viruses. The STAR enters the plant cell, right? Upon entering the plant cell, it will encapsulate the virulent particles. So it will surround, it will cover, it will encapsulate them, it will cover them up, seal them up. It reduces the separation of the virus particles in the protein and nuclear particle. Once this is achieved, the absence of food causes the virus to become inactive. So as I said before, it cuts them off. When it cuts them off, it renders them inactive, dormant, and that's how it controls it. STAR virucide will then open the conductive tissues that were blocked, allowing for movement of the virus particles and plant cells to start recover. So it will move them out of the way and allow the plant to recover now. So the plant can resume its normal operations and carry out the functions which you need it to. Now, growing up, I've always heard the saying that says, many hands make work light. So using the STAR virucide is excellent for your viral control, yes, but it can use some help as well. And the help can be in the form of the high yield package. The high yield package has antioxidant properties, vitamins and minerals. And what the high yield package can do to help the STAR is that it can boost the plant system, can promote the root and shoot development, because remember the plant was under stress when it was infected with the virus. And if it was not infected with virus and they're using it for a preventative measure, the high yield package is still applicable, as it will aid the plant in becoming more tolerant to the viruses when they come, right? So it will promote shoot and root development. It will improve branching and fresh growth, so you can have the plants bouncing back from the infection. It will include the pest and disease resistance, induce flowering, provide nutrient flow to flower. And you know, flower equals fruit, and it will prevent flower drop. It will increase the fruit size and quality and improve shelf life. So you can see that it works hand in hand with the STAR to improve your crop. Now the STAR has two actions. It's preventative and it's curative. Now how it will go about this is, if you are already infected with a virus, then you're going to have to go with the curative action. To do that, you'll have to make two applications relatively close together. So you'll have to spray today, and then 4 days later, you'll repeat the spray again, right? Now, after that 4-day interval, then you're going to put it on a 10 to 15-day spray interval. If you're doing it as a preventative, then you will do a 10 to 15-day spray interval from the get-go. and that will give you a preventative approach. Now the re-entry interval is 24 hours and the pre-harvest interval is 3 days. The application rate is 1 liter per hectare, or if you're doing it by the gallon, it's 3 to 5 ml per liter. There's a typo there. It should be per liter, 3 to 5 ml per liter, or 2 to 3 teaspoons per gallon. Now, of course, we here at Newport-Fersan practice bringing quality to the market. And one of the ways we do that is through our R&D division. So everything that we put on the shelf, we first test it to ensure that we bring quality to our farmers. So we, of course, put the STAR Virucide through rigorous testing, and we were pleased with all the results. The farmers are overjoyed, and thus we had to bring it to the market so that we can provide our farmers with the best technologies to boost their operations. Now you can see here on your right, the treated field versus the untreated field, and it's a day and night appearance right there. You have lush vegetation on your right, and you can see the difference in terms of productivity happening right there. Now, of course, something can look good and it's not performing. So we did not just only look at the aesthetic improvements, but we also look at the yield comparison between them. So in December 2017 on that field, you can see the bar chart showing you the difference. A quick calculation will tell you that it's in the ballpark of 200 pounds difference. Then you'll have 180 pounds difference in December, and January you will have around 100 and something pounds difference. And anybody can tell you those are not small figures for any operation. And the more you have in terms of poundage, will translate into more money in your pocket, right? We've reached the end of our presentation.
Now, as we move into our question and answer sessions, we ask participants, should you have a question, just raise your hand, use the raise hand feature on the platform, and you will be identified and give the go ahead to ask your question. All right, Mr. Kingsley Palmer, you have a question. You can go ahead and unmute yourself.
Yes, good night. Excellent presentation from what I heard so far. Question is there, if a farmer overuses the product, is there a possibility of any sort of resistance to the virus, by the virus, sorry.
Mr. Henry, you would like to take that one?
Mr. Palmer.
Yes, so, good night, Sir Palmer. And members. So you're asking if there's the possibility of resistance being developed. Based on the mode of action of the virucide, STAR Virucide, there's not the possibility of resistance being developed. It operates internally on the viral particles, but just imagine a similar operation as how, for example, an oil operates, either as a desiccant, drying out the organism, or a suffocant, suffocating the organism. To that, there can be absolutely no resistance. So in like manner, the STAR Virucide, once it encapsulates a particle, a viral particle, that's it. And there's no chance of the organism or the different strains of viruses developing resistance in this sort of operation.
Thank you, Mr. Henry. Ms. Kamalita, you can go ahead and unmute yourself and ask your question. Kamalita Starchan, you're still there? All right, Mr. Henry, there is a question in the chat that says, what viruses affects oranges?
What viruses affect oranges? For example, there is the Tristizia virus that affects oranges. I think, if I'm not mistaken, it was listed by Sir Jones. So that's one of the major, in fact, the major threat to orange production globally. In fact, it would have decimated a number of countries that would have been leaders in the production of. So there were different mechanisms developed to combat that. Separate and apart from that, as was discussed earlier, once there is the, a lot of plants are not, what I would say, a lot of viruses are not plant specific. So you will have different symptoms being seen are appearing. So for example, you might have cases where you might have the splitting, and you will have the uneven ripening, which are factors or symptoms of the Tristizia. There are some other viruses that the way they impact the plant is they actually cause the cracking of the bark and stuff like those. So yes, there are several viruses that affect us, but the major of them is the Tristizia, which is present and would have been the limiting factor globally.
Thank you, Mr. Henry. Mr. Denton Jr. Nation is asking, how is it treated if it is with the same star?
Good evening, Mr. Nation. Yes, the STAR Virucide can be used to treat this. And if it is already infected with this virus, then you're going to want to take the curative approach and then continue with the preventative approach. And just to remind you, that would be an application at day 1, and then 4 days prior to that, you'd apply it again. Then you can move to a 10 to 15 day spray rotation. And I think I saw in the chat where someone was asking for the contact information for Manchester. So Manchester falls within zone 3, and those are the contact information on your screen now. Mr. Donovan Price is your agronomist in charge of research and development, and his information is there. Ms. Claudine Smith is your technical sales consultant. Ms. Diane Walker is a telesales representative.
Okay, thank you, Mr. Jones. Ms. Kamalita, you can go ahead and quickly ask your question. Kamalita, are you hearing me? All right, we not seem to be getting anything from her.
Good night. Thank you. Hello.
Yes, we can hear you. Go ahead with your question.
All right. Question number 1. The information tonight, would it be sent like the presentation?
Good evening. Unfortunately, our policy does not allow for us to share our training material, but the good news is that we'll have a rebroadcast of this presentation this Friday at 7 p.m. So we'll rebroadcast the presentation. If there is still need for you to have further clarification on it, you can always reach out to us via any of our representatives as per your zones, or it doesn't need to be by zone. We can always direct it to the relevant personnel. Just contact us and we'll have that information passed on to you.
Okay, question number 2. What can be used to treat nematodes apart from your new product coming on the market? What other product is there that FERSAN provide for that purpose?
At the current moment, we do not stock anything in our inventory that is directly used for nematodes.
Okay, question number 3. What product can be used to boost the soil from deficiency?
Okay. Let me take that one, Sajae. Thanks. So if it is that we have established that there's a deficiency, it simply means that there would have to be a soil analysis done, because that's the only way to empirically determine what exactly is there and in what quantity. Once that is determined, what we do here at Newport-Fersan is to design what we refer to as a precise nutrient management program, where we seek to supply each nutrient element as per the needs be. So if, for example, we have a case where you have an excess or very high readings of a particular nutrient, irrespective of the nutrient and how important it is, as they all are, then you'll not get a program indicating that you need to buy that particular nutrient. Separate and apart from that, there are other factors that contribute to how well or productive your plants are, because in actual fact, soil fertility, despite the name as in fert, is not about the presence or the lack thereof of fertilizer. Soil fertility has to do with a balanced environment, meaning that what is required chemically, what is required biologically in terms of organisms, and what is required physically in terms of structure and other components of the soil is actually intact. So one of the things that we also look at when we do a soil test is the pH of the soil, because if the pH of the soil is incorrect, is too acidic or too alkaline, you could apply nutrients until, as they would say, the cows come home. You will not see the sort of development, growth and development and productivity of your crop. So one of the things, the first thing that we look at when designing a nutrient management program is basics. So we go to basic, which is what is the existing pH? Once there's need for that to be rectified, we have in-house at Newport-Fersan the necessary material. If the pH is too acidic, we can fix that. If it's too alkaline, we can fix that. And thereafter, we talk about the application of nutrients.
Okay, thank you. I was going to ask what's the cost for the STAR product.
I can answer that one, Mr. Henry. The cost for the STAR product per liter, that is $3,130, right? And should you venture in purchasing more than the 1 liter, if you purchase it by the case, we do have case prices where if you get by 10 liters, you can get it at a different price.
Okay, thank you. What is used to treat leaf roll? Well, STAR, of course, but other products from STAR.
So leaf roll, if it is that the causal agent is a virus, there's nothing else available that respond to a virus in the manner that STAR does. So STAR is the only known virucide at this point in time in the island. Yeah, so that's your go-to in terms of a virucide. And remember, Sajae would have pointed out in the management practice that one of the things that you also need to pay close attention to is how you control the possible vectors that transmit the virus. So your insects, your mites, and such the like.
Okay, I was going to ask the weed, like your Spanish needle, the whiteheads, and other various types of weeds. I know that they are the host for those type of insects via viruses because the insect transfer the virus. So the STAR would be the best product. So, well, I'd have to destroy every weed from the area which I'm going to plant. That's correct.
Well, the reality is, as you're rightly stating just now, is that weeds being plants are potential host plants. Yes? And it is for more than one reason it serves you best that a plant growing where it's not wanted, the definition for a weed, is actually removed. And you do as best as you can to ensure that the population of weed, and as I said before, for a number of reasons, because they compete with you for nutrient and water, and more often than not, they're more efficient users of some of the resources than your actual crop. So yes, your best effort will be required.
For what diseases causes blackleg? Like down at the end of the plant, near to the root, you see that black in there.
Okay, so you can actually have, and I would have looked at, I think it was in the Irish, the potato earlier, you could have more than one causal agent of a particular disease. And some of these are actually fungal, meaning it's caused by, as the name suggests, a fungus, or you could actually have bacterial infections. Now, once you start to have the deterioration of the cells, the tissues of the plant, that is normally one of the general signs of a decay. So to go off on a tangent to say this is or that isn't would be premature. What we normally like to do is to observe, take samples, do observation, and we have a capable unit at the Bozos Research Station that we call upon from time to time, right across the length and breadth of Jamaica, to isolate and determine what the causal agent of a particular disease actually is.
Okay, I have two questions left. What are the main border crops to be planted? I know that corn is one.
Okay, so corn, as you rightly said, is one. I would have had, and as recent as 2 years ago, I had a farmer doing hot peppers, large acreage, and we had to subdivide fields because of a number of reasons. One, we were having crop establishment at different stages, and you would understand and appreciate well that the susceptibility of your younger plants coming down with any pathogen is higher when compared to your plants that have developed fairly well. The older the plant, the younger the plant, the more susceptible or vulnerable they are. So what we did was to put in fever grass, lemongrass as border, and they were actually planted and they came up and were well manicured and look like a biological wall. And that worked well in preventing the transfer. So that's what's another crop that can be used as well.
Okay, and my last question, what is the cost of your high yield package?
That's Letitia.
All right, so the IEL package is for $4,330. And it
4,000.
can, right, $4,330. It can do 1 acre, right? It comes with 3 sachet package in it, which is used as at 3 different stages of your crop cycle, right? And once you obtain this product and you're ready to use the product as well, we do have agronomists and team members available that will assist you in walking you through with the product.
Okay, thank you very much.
You are most welcome. And let me say to all the participants here with us tonight, we do appreciate you taking the time to log on with us tonight and sharing in this presentation, right? Thank you, ladies and gentlemen, farmers, for participating. Thank you to Mr. Henry and Mr. Jones for that wonderful presentation. Do have yourself a wonderful rest of the evening.